Hello, my name is Chatushika Samradivakara and today I'm going to do a presentation about role of the pancreas and liver in regulating digestion. This is my full name and today's date. Let's talk about the liver first. Liver. It is the largest internal organ in our body and it weighs 3.3 pounds in an adult. It lies just under the diaphragm towards the right side of the body. As you can see in the picture, this is the anterior view of an liver and this is the posterior view of an liver. Hepatocytes of the liver organized as irregular hexagonal units as lobules. Blood entering the liver brings nutrients and foreign substances from digestive tract. Bilirubin from hemoglobin breakdown and metabolites from peripheral tissues of the body. When we talk about the liver in regular digestion, the main point is the secretion of bile. Bile is a non-enzymatic solution secreted from hepatocytes. The key components of bile are bile salts, bile pigments, and cholesterol. Bile salts facilitates enzymatic fat digestion. Bile pigments such as bilirubin, which are the waste products of hemoglobin degradation. Cholesterol is excreted in the feces. Bile secreted by hepatocytes travel in hepatic ducts to the gallbladder, which stores and concentrates the bile solution. During a meal that includes fats, contraction of the gallbladder sends bile into the duodenum through the common bile duct. The bile salt are not altered during fat digestion. They recycle back to the liver. It's an essential part of fat digestion. The intestinal, pancreatic and hepatic secretion of enzymes and bile is essential for normal digestive function. Pancreatic and brush border enzymes do the digestion of peptides, carbohydrates, and fats into smaller molecules that can be absorbed. Fat digestion is complicated by the fact that most lipids are not particularly water-soluble, but bile salts are like phospholipids of cell membrane. The hydrophobic regions of the bile salts associate with the surface of lipid droplets while the polar side chains interact with water. It creates a stable emulsion of small water-soluble fat droplets. As you can see in this picture, there's the liver, gallbladder, pancreas and the bile duct, which I was talking about. The enzymatic fat digestion is carried out by lipase. It removes 2 fatty acids from each triglyceride molecule. That's 1 monoglyceride and 2 free fatty acids. An enzymatic and mechanical digestion process, fatty acids, bile salt, mono and diglycerides, phospholipids and cholesterol coalesce to form small disc-shaped micelles. Now let's move to the pancreas. Here's the picture of a pancreas, which is an endocrine and exocrine organ. Endocrine secretions come from the islets and it includes the hormone insulin and glucagon. Exocrine secretions include digestive enzymes and a watery solution of sodium bicarbonate, and it is consisting of lobules called acini, and acini cells secrete digestive enzymes and the duct cell secretes the solution. Let's talk about the pancreas in regulating digestion. There are 2 main parts in the pancreas in regulating digestion. The first part is the enzyme secretion and the second part is about the bicarbonate secretion. When we talk about the enzyme secretion, most pancreatic enzymes are secreted as zymogenes that must be activated upon arrival in the intestine. This activation process is a cascade that begins with brush border enteropeptidase converts inactive trypsinogen to active trypsin. Trypsin then converts the other pancreatic zymogens to their active forms. The signals for pancreatic enzyme release include distension of the small intestine, the presence of food in the intestine, neural signals, and the GI hormone CCK. Pancreatic enzymes enter the intestine in a watery fluid that also contains bicarbonates. When we talk about the bicarbonate secretion into the duodenum neutralizes the acid entering from the stomach. A small amount of bicarbonate is secreted by duodenal cells, but most comes from the pancreas. Bicarbonate production requires high levels of enzyme carbonic anhydrase. Bicarbonate produced from carbon dioxide and water is secreted by an apical chlorine bicarbonate exchange. Hydrogen ions produced along with bicarbonate leave the cell on basolateral sodium-hydrogen exchanges. The hydrogen thus reabsorbs into the intestinal circulation, helps to balance bicarbonate put into the blood when parietal cell secretes hydrogen ions into the stomach. And that's all about the regulating digestion in pancreas and liver. And let's move to my assignment. It's about the fat digestion and absorption. So I want to do the terms, which means the defining the following terms, and if a diagram is included, that would be nice. All the terms you can be seen in here. There are 5 terms. The first one is the emulsification process of fat digestion. Second one, fat hydrolysis in fat digestion. Lipid uptake by micelles and chylomicron formation of fat absorption and chylomicron exocytosis and lymphatic uptake of fat absorption. So the first term can be described as fat globules is broken up and coated lecithin and bile acids. Where the fat hydrolysis in fat digestion is the emulsification droplets that are acted upon by pancreatic lipase, which hydrolyze the first and the third fatty acids from triglycerides, usually leaving the middle fatty acid. And the third one, lipid uptake by micelles, is that the micelles in the bile pass to the small intestine and pick up several types of dietary and semi-digested lipids. Fourth one is can be described as the intestinal cells absorb lipids from micelles, resynthesize triglycerides and package triglycerides, cholesterol, and phospholipids into protein-coated chylomicrons. And the fifth one, the chylomicron exocytosis and lymphatic uptake of fat absorption, that's can be defined as Golgi complex package. Chylomicrons into secretory vesicles. Chylomicrons are released from basal cell membrane by exocytosis and enter the lacteal of the villus. And a diagram like this can be included. As you can see, this is the fat globules and this is the emulsion droplets, and it is digested by lipids. Then it becomes free fatty acids and bile salt. Then how it goes to the micelles and in the epithelial cells, what's the process and how it goes into the lacteal. Finally, that would be the assignment, and I think that's about it. Thank you.